right, good morning, everybody. Today is Monday, June 4th, 2018, and this is Before the First Cup of Jules here on UCY.TV. Happy Monday, everyone. It is a soggy and overcast 66 degrees, but feeling very green and plush here in upstate New York. So where we leave off on Friday, we titled the show Food, Phages, Autoimmune, Viruses, Chronic Ills, and Cancer. And we had taken a look at some of the connections between viruses in our environment and getting infected with different viruses, whether they be known or unknown, and some of the viruses that can live within your DNA for 30 years without you even having a clue until they reactivate. Things such as shingles, though we didn't talk about shingles, but the Varicella virus is a DNA virus, as are the herpes family of viruses that include many things such as Epstein-Barr. We also looked at a bovine leukemia virus that they've known about since the 70s in our milk that they have now found in breast cancer. We also, I think we made it to looking at Epstein-Barr and chronic fatigue and autoimmune and thyroid disorders. And it also can trigger some sort of brain issues, whether it be what they call foggy brain or actual blood flow issues by affecting your arteries. There are a lot of things that viruses can do to the human body. Now, we also discovered that they are using phages as a new line of defense against antibiotic-resistant bacteria, which, of course, is now a crisis. Obama had come out and talked about one of the federal edicts that they were going to go after, and that would be coming up with a solution to antibiotic-resistant bacteria. And one of the things that we don't typically think about is how the interplay between these very small snippets of life, these very small snippets of DNA from these other organisms, can potentially influence our evolution as a human species, because we are, I hate to use the term infected, but we are infected in our guts especially with all kinds of different phages as well as different bacteria, and that would be our microbiome, our virome, and then there's also another thing called the metagenome in our guts, and we're going to look at those once we make it through this long progression of stories today. And this is going to be a setup, by the way. I'm doing this special edition on a Monday. Since January, I've been skipping Mondays and giving myself a long weekend, but doing this special edition on a Monday because hopefully, cross your fingers, on Wednesday I do have confirmation from Clint Richardson. He's going to be on, and we have to get in contact with Patrick Jordan, see if we can get him on as well. And we are going to actually look at some of this co-evolution that is happening between parasites and humans on this planet, and how potentially this could be accelerating our evolution, and maybe it's even self-directed evolution by mad scientists. All right, so let's start out here. Here's a story I found out of phys.org, May 25th, 2018, so about a week ago, titled The World of Parasites. It's just so lovely. Alex Betts, Craig McLean, and Kayla King from the Department of Zoology shed light on the recent research published in Science, which addressed the impact that parasite communities have on evolutionary change and diversity. It has been over 150 years since Darwin published The Origin of the Species. Nevertheless, the evolution and consequences of biological diversity are pressing questions that are still being tackled by evolutionary biologists. The world is full of parasites, and their antagonistic interactions with hosts can have big evolutionary consequences. Virtually all organisms are infected with parasites or are one themselves, and heavily infected hosts can be under strong natural selection as they are unlikely to reproduce. Hosts can reciprocally impose strong selection on parasites as resistant hosts can fight them off, crippling parasite ability to establish an infection and reproduce themselves, such as a virus. And this intense mutual antagonism can drive rapid evolutionary change, ding, 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 and is thought to be possible for many other aspects of biology, from sex to speciation. Hosts are often under attack from multiple different parasite species, and we tested whether this parasite diversity shapes the mutual antagonism with hosts over evolutionary time, i.e., coevolution. The research published today in Science, we studied interactions between single-celled bacterium such as Pseudomonas aeruginosa, which I've never heard of that one, which acts as a host for bacteriophage parasites, ding ding, which are tiny little viruses that infect bacterial host cells, reproducing inside of these cells and burst their host to release their progeny within the organism. This host parasite system is very useful because we can observe coevolution between pseudomonas and phages in real time. And at the start of the experiment, the bacterial clone we used was susceptible to all of our viral parasites. However, bacteria rapidly evolved resistance to phages again. This might not be fixing our quote-unquote antibiotic-resistant bacteria problem. This might make it even worse because they can rapidly evolve resistance to phages by modifying their cell surfaces to make it harder for phages to attach and infect. Phages must attach to the cell surface before they inject their lethal DNA, and in response, phages evolved modified structures to attach to the altered cell surfaces. Diverse parasite communities are typical in many ecosystems, and remember, people, that's one of the big deals about all this mad science. They are letting these genies out of the bottle, and we live in one big ecosystem. So if they decide to implement a gene drive in, let's say, New Zealand and wipe out all the rodents because, they didn't naturally come from there, they came on ships, well, how is that going to affect the wildlife that is living in New Zealand, let's say? Because as soon as they get rid of mice and rats, where are the hawks going to get food? I mean, maybe the ocean, but that might be a little more difficult than catching a little mousey. So anyway, our work shows that relative to infection by a single parasite species, parasite diversity can drive even more rapid evolution in host resistance. Host populations also become more evolutionary divergent from each other. Ultimately, this genomic split between populations could lead to a formation of a new species of bacteria and phage. So we're actually moving new life in the viral and the bacterial species of what is inhabiting our planet. Now let's add a little bit of insult to injury. I love to rub salt in the wound, but this one caught my attention as well. 2 days ago out of phys.org, June 2nd, 2018, it's titled, Team Discovers How Microbes Survive Clean Rooms and Contaminate Spacecraft. Now, if you remember, we looked a while ago at them sending MRSA up on, I think it was on one of the SpaceX missions that was helping to resupply the international space stations. And they sent MRSA up there to work on an experiment to make MRSA more virulent, because in space, viruses and bacteria tend to become more virulent. And I was also reading that space depresses the immune system of astronauts, which can make things a little more virulent in space. But listen to this. Team discovers how microbes survive clean rooms and contaminate spacecraft. Rashik Mogul, a Cal Poly Pomona professor of biological chemistry, was the lead author of an article in the journal Astrobiology that offers the first biochemical evidence explaining the reason that contamination persists. Chemistry professor Gregory A. Barding Jr. was a collaborator and a second author on the paper, and the remaining 22 co-authors, and we're not going to list them here, We're all chemistry graduate students and undergraduates in biological sciences. We designed the project to give students hands-on experience, blah, 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 blah. In the clean room facilities, NASA implements a variety of planetary protection measures, yes, planetary protection measures, to minimize biological contamination of spacecraft. These steps are important because contamination by Earth-based microorganisms could compromise life detection missions and provide false positive results. Despite our extensive cleaning procedures, however, molecular genetic analyses show that the clean rooms harbor a diverse collection of microorganisms or spacecraft microbiome. Did you know spacecraft had a microbiome? And that would include bacteria, archaea, and fungi. The Anciobacter, which is a genus of bacteria, are among the dominant members of spacecraft microbiome. And to figure out how spacecraft microbiome survives the clean room facilities, researchers analyzed several Ancetobacter strains and were originally isolated from the Mars Odyssey and Phoenix spacecraft facilities. They found that under very nutrient-restricted conditions, and this sounds pretty scary to me, most of the tested strains grew on and biodegraded the cleaning agents used on spacecraft during assembly. Work showed that cultures grew on ethyl alcohol as a sole carbon source when displaying reasonable tolerances toward oxidative stress, like a super bacteria. And this is important since oxidative stress is associated with desiccating and high radiation environments similar to Mars. Uh-oh, we just sent a plague to Mars. The tested strains were also able to biodegrade isopropyl alcohol as well as Cleanol 30, which are two other cleaning agents commonly used, and these products potentially serving as energy sources for the spacecraft microbiome. We're giving the planetary protection community a baseline understanding of why these microorganisms remain. There's always stuff coming into our clean rooms, so maybe they need a flamethrower. But one of the questions has been, why do microbes remain in the clean rooms? And why is there a set of microorganisms that are common to clean rooms? For planetary protection, this indicates a more stringent cleaning setup needs to be used for missions focused on life detection, but we don't care about contamination, and highlights the potential needed to use differing and rotating cleaning reagents that are compatible with spacecraft to control the biological burden. Now, my question is, what about the reverse? What are they doing with shit they're bringing back from space missions? What are they doing with spacecraft that re-enter the atmosphere? And I know they usually have a good burn up there, but I mean, they bring things back. They do rock samples, supposedly, wink, or if not yet, they're going to be doing mining missions and bringing back samples of different elements, precious elements to this planet. How are they planning on decontaminating that to make sure that they don't bring back any super microorganism that we don't know about? Well, don't worry about it, because the World Health Organization has just recently simulated a global pandemic, and it happened to leave 150 million people dead. A real one could be much, much worse, and I think Billy Gates was talking about 300 million. This was published May 31st, 2018. Out of the South China Morning Post. Exercise simulated a global health disaster with fictional elements to force government officials and experts to make the kind of key decisions they could face in a real pandemic. A novel virus, moderately contagious and moderately lethal, has surfaced and is spreading rapidly around the globe. Outbreaks first appear in Frankfurt, Germany, and Caracas, Venezuela, and the virus is transmitted from person to person, primarily by coughing. And remember, we had read about them predicting that the next pandemic might be an airborne one. There is no effective antiviral or vaccine, and American troops stationed abroad are now infected. But the first case to reach the United States has been identified on a small college campus in Massachusetts. Well, go figure. Probably working with the likes of Georgie Porgy Church. So began a recent day-long exercise hosted by Johns Hopkins Center for Health Security, and the simulated mixed details of past disasters were with fictional elements to force government officials and experts to make the kind of key decisions they could face in real life. It was a very tense day. The exercise was inspired in part by the troubled response to the Ebola epidemic of 2014, and everyone was acutely aware of the very real and ongoing Ebola epidemic that is currently spreading in the Congo. In the simulation, a bipartisan group of current and former high-ranking U.S. government officials played a team of presidential advisors faced with a host of real-world policy, political, and ethical dilemmas, like whether or not to test some of these newfangled vaccines on everybody that's in quarantine. The actors included former Senate Majority Leader Tom Daschle, who played the Senate Majority Leader, and Representative Susan Brooks, who actually played herself. They had to react as the outbreak unfolded according to a script provided by Johns Hopkins with no advanced knowledge about how the mock disaster would play out. And I've actually been a part of one of these events where they give you a situation and everybody has a role and they just give it to you and you all sit around a table. It's called a tabletop exercise. They face difficult questions throughout the exercise. Should the United States impose a travel ban on flights from Germany and Venezuela? I'd probably say so. And if not, they face the problem of explaining that decision to the American public and Congress in the face of political pressure to act. Should the United States send troops to Jordan, where a major outbreak has occurred and key Middle East allies are requesting assistance? I wonder if that was Saudi Arabia. Once a vaccine is developed, who should get it first? I would say send it to Africa because that's kind of par for the course. And or maybe India, or should priority go to officials to ensure continuity of government or to children and pregnant women? I remember in the exercise that I worked on, they told us that if we were working on the actual process of getting prophylaxis out to the public, and that would be whether you're handing out antibiotics or doing some sort of clinic to give vaccines, that the people who were manning the clinics and their families were going to be the first to get it. So that was pretty frightening. Or should there be a lottery to see who gets it and who doesn't? There were no easy answers, and that was the point of the day-long exercise, held in a darkened hotel ballroom before a wrapped invitation-only audience of about 150 people and live streamed on Facebook. Oh, I should see if I could find that video because that'd be pretty interesting. There were experts from academia and think tanks and officials from across the U.S. government, including the White House, defense and intelligence communities, health and security agencies, as well as Congress. Tom Inglesby, the director of Johns Hopkins Center, says that the purpose for the exercise was to provide experiential learning for new decision makers in the Trump administration. But many of these hard issues have remained unresolved for several administrations, except for the Bush administration. Just give them all experimental vaccines that we're putting in the stockpile and slippery palming all of our buddies in Big Pharma. There's still a lot of moving pieces in the world of pandemic preparedness at the moment, he says. And chief among those issues are questions about the extent of U.S. support for global health issues and the role of national security communities. Even though many players in the scenario had decades of experience in health security and national and global health policy and law, there were still so many unanswered questions and capability gaps. Cameron, who was in the audience, is Vice President for Global Biological Policy at the Nuclear Threat Initiative. Well, that's kind of an interesting title. Global Biological Policy at the Nuclear Threat Initiative. In the fictional outbreak, one or even one less deadly emerged tomorrow, she said. We'd be facing a scenario with a new cabinet untested by a major outbreak. Unlike Ebola, which spreads through direct contact and bodily fluids, this Clade X virus, which was this hypothetical virus in this tabletop exercise, in this Johns Hopkins simulation, was a flu-like respiratory virus which would spread far more easily from person to person through coughing and sneezing. This fictional outbreak kept getting worse. It had a 10% fatality rate, about the same as SARS virus that traveled around the world in 2002 to 2003. And because the virus in the drill was new, no one had previous immunity, and it spread quickly in large cities. It killed more than 100 million globally. The health care system collapsed. Panic spread, and the U.S. stock market crashed. And the president, members of Congress, and the Supreme Court were all incapacitated. We didn't want it to have a Disney-like ending, says Inglesby. We wanted to have a plausible scenario, and we knew it would be jarring. Oh, no. So the government was all taken out in it as well. And over the course of the day, 10 experts played the roles of U.S. officials in a series of simulated National Security Council convened meetings. They acted the parts of National Security Advisor, the Secretaries of Health and Human Services, of State, Homeland Security, and Defense, Attorney General, and the directors of the Central Intelligence Agency and the Centers for Disease Control and Prevention. There were also 2 members from Congress and a National Security Advisor. The fictional outbreak was revealed to be a virus engineered, ding-a-ling, in a Swiss lab by a terrorist group. Another grab from the headline's twist, the terrorists had inserted deadly genes from the Nipah virus, which is a rare brain-damaging virus. And actually, if you allow me to pop ahead 1 second here, let's take a look at the Nipah virus. Just got to load. Deadly Nipah virus. This is that one I mentioned out of India, which can harm the brain. They are experiencing an outbreak in India, May 30th, 2018, and the outbreak is still ongoing. The outbreak of a virus with deadly effects on the brain has killed 14 people in the southern Indian state of Kerala, setting off worries about its potential spread. The Nipah virus can lead to inflammation of the brain, resulting in a coma, which is fatal 40 to 70% of the time, according to the World Health Organization. Centers for Disease Control and Prevention says it is transmitted through infected bats, which I saw an update to that. They cannot find the bat who is bat zero, as well as pigs and from other infected people. According to the World Health Organization, there is no effective treatment. This current outbreak was first reported earlier this month in India's southwest coast, and officials have confirmed now 16 cases with another 12 suspected, and the latest death occurred this week. According to an Indian newspaper, 4 individuals in one family died in a village called Changarath, more than an hour north of Kazahingrod, and as many as 90 families there and in 2 neighboring villages have now left their homes. People have been avoiding the fruit region because of fruit bats. Remember, that's how they say Ebola started, with the original transmission believed to have come from a fruit bat. Now, why, you ask, do they keep talking about fruit bats and infections? Well, we're going to take a look at that. If you give me 1 second here, let me just roll over a few tabs and see if we can find that. Yes, here we go. Bats are blind to the deadly effects of viruses. I don't know if you guys knew this. I taught my daughter something new yesterday, little Miss Smarty Pants. Humans infected with a flevovirus, such as the Marburg virus, which is a hemorrhagic type fever, can face severe illness that can include uncontrollable bleeding, high fever, multiple organ failure, and in many instances, death. Well, I would imagine so. Now, hold on, because we're going to move this one over too, because this will be the next one. Well, let's go back. Okay. A natural reservoir for the Marburg virus is the Rosetta's aegyptius, which is the Egyptian fruit bat. An intriguing detail regarding bats infected with a highly lethal virus is that they are unique in their hosting duties, and that would be infected bats don't get sick. It is thought that there are differences in the immune system of bats relative to other mammals, and that underpins their ability to control insidious intruders. Specific details, however, have been lacking, so bats are going to be ripe for some mad science laboratory experiments, they're saying. And Palkovich and other co-authors used a hybrid short and long read sequencing strategy to generate a high-resolution draft genome of the Egyptian fruit bat. It is the most contiguous bat genome assembled to date. So they're now assembling the bat genome. Their investigation gave considerable attention to the study of immune-related genes, given their unique response that the bats have toward a viral infection. Now remember, they also just recently kicked off a synthetic biology project looking to make virus-resistant supercells that involve Georgie Porgy Church. I don't think that these things aren't all connected. So let's get back to the bats. So they sequenced the biggest contiguous bat genome, and their investigation gives considerable attention to the study of immune-related genes, giving the unique response that these bats have toward viral infection. The research team observed conspicuous evolutionary positive selection and a large expanse of immune gene families, including major histocompatibility complex class 1 genes. Natural killer cell receptors were also expanded, and that's something that is actually taken out or suppressed in some of those infections with Epstein-Barr that we had looked at on Friday. The natural killer cells have issues, but in bats, natural killer cell receptors were expanded and remarkable in that they displayed both activating and inhibitory interaction motifs. Transcription suggested that several natural killer receptors with inhibitory signaling characteristics were expressed at elevated levels, meaning that they go into overdrive and take out any type of detected infection. So now let's pop a little bit forward, because I don't want to get too technical. I want to get to kind of the end result that's going to fan into Wednesday's new show. This one is titled, and we have talked about entomological issues before with Lyme's disease and Plum Island and infection that ensues with a bacteria, which is a type of spirochete that gets into the body, and it can hide in your joints for many, many years until it is reactivated. Ticks have been a vector for disease for a very long time. Well, listen to this one. Just like on Plum Island and how they suspect Lyme disease got off the island by ticks jumping onto migratory birds, well, there is a new migratory bird tick-mediated virus. Ticks on migratory birds out of Medical Press, medicalexpress.com, June 1st, 2018. Ticks on migratory birds have been found to carry a newly discovered hemorrhagic fever virus. Oh, look out. In a brand new study, researchers at Uppsala University and other institutions have identified genetic material from a recently identified Alzheimer's hemorrhagic virus in the tick species Hyaloma rifilpes. The discovery was made after thousands of ticks collected from migratory birds that were captured in the Mediterranean basin were part of the study, and the results indicate that the birds could contribute to spreading this virus to brand new geographical areas. We should probably find out where these birds migrate to and from. The Alzheimer's virus was first found in Saudi Arabia in the mid-1990s. Well, the country and the timing of that is pretty interesting. In patients with typical hemorrhagic fever symptoms. The conditions resemble diseases like Ebola, but seem to be spread by contact with camels and cattle or from tick bites that are not directly between humans. The knowledge about this relatively novel virus is still limited, and it is not exactly known which animals can act as hosts for the virus and which insects or tick species will actually transmit the virus. But previous research has pointed to camels and other domestic livestock as potential carriers of the virus, and one of several tick species possibly spreading the disease between mammals, including humans. The virus is close to a relative of another tick-borne virus, which is the Cancerforous disease virus, also found in India. And so far, the Alchemira virus has only been seen on the Arabian Peninsula and in multinational collaborations. Researchers at the Uppsala University have detected the Alchemira virus RNA in several ticks that were removed from birds migrating from Africa to Europe. Uh-oh. The purpose from the study is to find out which tick-borne diseases that could potentially be spread to new regions with migratory birds, and thousands of ticks were removed from migrating birds at several different bird observatories in the Mediterranean basin. So, all right, now let's go to a publication that comes out of Nieman School at Harvard University. What is interesting about Harvard University is that is actually where Georgie Porgy Church is residing and a lot of the people who happen to work with him. This is an interesting publication titled Covering Pandemic Flu, Understanding How Flu Viruses, so not just any virus, not just a hemorrhagic virus, but this is a flu virus which is easily transmitted usually between human to human and can sometimes even become pandemics by first transferring from animal to human and then human to human. Flu viruses have the capacity to both slowly, through small genetic changes that are passed down to daughter generations, and quickly through a process called reassortment that mixes larger genetic segments from several virus strains to create a new virus. Both processes are important to an influenza's success as a disease-causing organism during flu seasons and in pandemics. And that means that they can evolve either slowly or quickly. This page provides an overview of what scientists have learned about basic mechanisms behind influenza viruses' ability to change, and also this selection will help you sort through the major terms used in the discussion, from genetic shifting to genetic drift to triple reassortment. I don't know if we're going to get that far. To understand major genetic changes that lead to a pandemic in a pathogen, it helps to first understand the minor genetic changes that produce new flu strains in each flu season. Like all living things, influenza makes small errors, or that would be mutations, when it copies its genetic code during reproduction. But influenza lacks the ability to correct those errors because it is an RNA virus. We talked about that the other day. RNA, unlike DNA, lacks a self-correcting mechanism, and DNA would have that second strand that serves as an error detect and correct system. And as a result, influenza is not considered genetically stable, meaning that it can mutate very frequently. And every generation is slightly different, like those differences, and those differences tend to accumulate as times pass. A slow deviation is called an antigenic drift, and is the reason why it is necessary to reformulate flu vaccines every single year. And every flu season, the genetic makeup of the dominant strain from the prior year will have drifted, changing the surface structure of those strains just enough to diminish or even destroy the effectiveness of the previous year's vaccine. And that would probably be because the receptor that they were targeting no longer exists or has changed shape. Each winter, health authorities must make an educated guess which strains are likely to dominate the next season, and it takes 6 or more months using eggs to develop and manufacture vaccines from those strains. But don't worry about it, because they're getting better with mad science. We're going to talk about that in a story or two. And in some years, genetic drift during those 6 months can render newly formulated vaccines ineffective, leaving the entire population vulnerable to a newly evolved virus. Guys, viruses are very frightening to me when you think about all of the mad science that is going on in the lab and some of the tinkering that they are doing to see what would happen, and even down not only long, dark hallways, but in mommy's back bedroom or in a rental garage in some outback community with a CRISPR kit. All right, so now let's continue on to another publication, HomelandPreparednessNews.com. Research develops framework to identify microorganisms with high pandemic potential, May 30th, 2018. This actually goes back to that tabletop exercise that we had talked about a story or two ago. Researchers at Johns Hopkins Center for Health Security recently developed a framework to help identify microorganisms that are most likely to cause pandemic and inform future preparedness and response efforts. The report from the researchers, called The Characteristics of Pandemic Pathogens, describes the framework and provides recommendations for improving global catastrophic biological risk, more affectionately known as GCBR preparedness. The health security community typically relies on historical examples of outbreaks, which does not account for biological agents that do not have a historical precedent or are not known. Roll out the experimental plasmid vaccine, because that's what's coming. This framework could help prepare for a global catastrophic biological event with no historical precedent. Guys, this is what they're prepping for, and these should be the things of nightmares. This is where I want to get myself a level 4 suit and a bunker. This is for events such as this that they are dangling out there as potentials, and maybe even stuff that they bring back on a space or mining probe that can't be killed in a clean room. Health security preparedness needs to be adaptable, and remember that these micropathogens can also have effect on our health, especially if they can get into our gut and start changing our natural microbiota and our virome, that then causes some sort of biochemical change in our brains that affects our behavior. I mean, I know Clint has talked about that before. There was a show on Netflix, I think, that was about this brain pathogen that got in and changed everybody. And maybe it got into their ear. That's what it was, this organism. And changed their brain and made everybody like really lefty leaning or something. And it was very funny, but it's not really funny because it is potentially a target. And what if somebody even on purpose engineered an organism that was able to do that? I mean, it frightens me. This is the stuff of my nightmares. And if I can stop this video that is about to play here, for an example, one of the things I wanted to illustrate here is this last flu that went around back in March, I believe, up in upstate New York at least. I think I had mentioned on the show that it hit me pretty hard, and I was still writing at the time, and I had noticed that I was having a lot of difficulty thinking, and a story came out a few weeks after I had started it to get better. There, I stopped the video. Shortly after I started to get better, to talk about that new flu strain that I had had and how they actually did find in research that it did have a very marked effect on the brain and your ability to remember things. But how does flu affect your mind? This is out of Newsweek.com, February 27, 2018. Bad memory and brain changes could result after your viral infection. I don't think this is just flu that does this. Even that Nipah virus in India is causing effects on the brain, and who knows what else. There was something called a panda syndrome that I had run across that they had discovered in adolescence when they got like strep throat or a sinus infection that caused swelling near the sinus and breathing cavities that go up in the front of the face that pushed on parts of the brain when they were faced with inflammation. And remember, any type of infection will cause inflammation. And so if it is something that can get near the brain, it may even cause some neurological effects. So did you get the flu this year? This is this Newsweek article. There's a chance that it may have had long-lasting effects on your brain. That'd be that brain fog. Researchers published Monday in February in the Journal of Neuroscience found that months after mice had gotten the flu, their memory was impaired. And for this study, mice were infected with a strain of the flu and put in something called a Morris water maze, basically a pool of water with submerged platforms that the mice can't see. And each time the mice got to the maze, they swam around until they touched the platform. But they gave them cues in the maze, like letters or colored bits, and they spread them around the pool. And after a few times out, the mice should learn where the platform is based on these cues, unless they have memory problems. Well, mice infected with the flu went through the test 4 times over 6 months, and for at least 2 months, they took far longer than was expected to find the platform. The test was done at 30, 60, 90, and 120 days after the infection, and at 120 days after the infection, the mice had shown that they had fully recovered. Now, that's crazy. They got back their full cognitive abilities, but you have to keep in mind that 120 days for the lifespan of a mouse would be equivalent to many years in the lifespan of a human. How nuts is that? This change may be because the brain's version of an immune system continues to be triggered long after the sniffles subside. The inflammatory response, ding ding, in the brain holds on much longer than the actual virus is around. 2 kinds of cells in the brain are responsible for this inflammatory response, and they would be your astrocytes and your microglia. Microglia is also thought to be involved in Alzheimer's as well as other conditions, and exactly how the virus might be having an effect isn't quite clear. But this isn't the first time that scientists have found evidence of an infection causing brain inflammation. There was a study involving a potential link between viruses and Parkinson's disease for years, and the findings actually support this paper, but any virus that gets into the brain becomes much more worrisome in terms of the later effects. So don't worry about that, because there is now new manufacturing technology platforms to speed pandemic vaccine development. June 1st, 2018, in a publication called InfectiousDiseaseAdvisor.com. Will the United States be ready for the next pandemic? And will the advent of platform vaccine technology vaccines can be developed quickly after the spread of infectious disease outbreaks? Remember, DARPA was already putting out a challenge to help control pandemic X in 60 days. So while traditional vaccines for non-epidemic diseases will take up to 15 years to develop, platform vaccine manufacturing uses efficient processes to establish from previous similar vaccines that can permit the start of phase 1 testing just months after the viral sequence selection of the pathogen. And as a result of this new platform, the time from viral sequencing to the pathogen to phase 1 trials of the vaccine has decreased from 20 months in 2013 for SARS to 3 months during the 2016 Zika virus epidemic. Notice how they are using these quote-unquote outbreaks that have happened over the last 10 years or so, or 12, 15 years or so, as ways to ramp up production and to evolve their method of production. So when the big one hits, something that they've been training for, they'll be prepared. If you are testing vaccines in the middle of an outbreak, then you can clearly get what is called vaccine efficacy signal pretty quickly. Yeah, so let's just test it on everybody and anybody because it is an outbreak, says Anthony S. Fauci, who is the director of the U.S. National Institute of Allergy and Infectious Diseases in the Pit of Lizards in Bethesda, Maryland, in an interview with Infectious Disease Advisor. If you are testing vaccines when there is a modest amount of cases, that is when it will take a few years before you get an efficacy signal. So let's just get a pandemic to get our testing done quickly. So when you get used to working with a particular platform technology, it is interchangeable with whatever virus you are dealing with because you have a plasmid, and we're going to talk about plasmids in a few minutes, ding ding, whose characteristics you know, and you can insert an appropriate gene into the plasmid because they're going to genetically engineer these to target whatever RNA or DNA they need to target. The FDA knows and understands the vaccine safety because you are already dealing with them with multiple different iterations of the vaccine. So it sounds like they're just going to rubber stamp it as they mix and match for different pathogens into the plasmid that then they infect you with, that then goes in and modifies your DNA. Another example of vaccine platform manufacturing are simian adenoviruses, and we read about those before. They have been able to put CRISPR-Cas9 into an adenovirus and then infect an organism, which are more immunogenic in humans. Variants of the simian adenovirus vaccine platforms were explored in 2014 during the Ebola outbreak and the high priority diseases that the World Health Organization has identified. Reducing the time to undertake clinical development is important, of course, and in many countries, the approval process for clinical trials is unnecessarily slow. So we have to circumvent that. We need a more streamlined approach on a global level to clinical trial approvals. In a multinational effort, the United States, along with the World Health Organization, continues their surveillance of the next major outbreak, and the World Health Organization has identified several priorities for vaccine research. One would be Crimean Congo hemorrhagic fever. Two would be Ebola virus and Marburg virus disease, and that would be like the one that is spread by those ticks. Third would be Lassa fever. Fourth would be the Middle Eastern respiratory syndrome known as MERS. Fourth would be SARS. Fifth would be Nipah and other henipapial diseases. Seventh would be Rift Valley fever. Eighth would be Zika. And ninth would be Disease X. Disease X is any unknown pathogen that is likely to be the world's next pandemic. And the World Health Organization acknowledges that this needs to remain nimble as priorities would shift Disease X from the existing known threats. Yeah, this is the experimental stuff. Along with World Health Organizations, other non-governmental organizations will be working on this as well, including a consortium of scientists, philanthropists, and investors that are not beholden to government or industry. Yeah. All right. So now let's look at a consortium of scientists. Let's look at, and again, I'm getting that loading slow problem. Come on, Paige. Let's go. I might have to sing in a second. I'm sorry. Come on, come on, come on. Do 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 do. Okay. I don't know if this. I don't know. Hold on. George Church has a wild idea to upend evolution. And here is your guide. So George Church has talked about creating a synthetic human genome. We talked about that ReCode project a few shows ago and a long time, quite a while before that. But he is also looking to bring back the woolly mammoth, all in the name of climate change. It's going to fix climate change. Remember I told you there was a story about that? Well, George Church has also looked at evolution and how we can do self-directed evolution. And one of the things he's working on is trying to come up with a way to make a synthetic project that would make us impervious to viruses. And I don't know. I can't find it in this story, so I guess we're going to have to put George Church on the back burner. So, all right, hold on. So as they've been tinkering with DNA, they've been doing all kinds of things. Let me go to the story. Actually, here's a few things that I want to talk about. So they are looking to cure us utilizing all of these different types of CRISPR technologies and changing different DNA shapes. This one was about this vaccine, DNA-based colorectal cancer vaccine is going to be tested in the first human trial. This was May 22nd, 2018. Potentially life expectancy increasing DNA-based colorectal cancer vaccine treatment is set to undergo its first human trial, according to research published at Digestive Disease Weekly 2018. Researchers in Melbourne, Australia, have tested a combination therapy of a DNA vaccine with an antibody that eliminated the body's natural defenses against the DNA vaccine. So they can even knock out your defenses to a DNA vaccine in several mice and force them to develop colorectal cancer. Tumors in the mice responded well to the vaccine, curing their cancer in about half of them, but the treatment of the mice were expected to live a few days, but that is 50% of them live for up to 2 years. I also saw that there was a sickle cell human trial that was supposed to be happening, and the FDA just put out some doubts, and they canceled the CRISPR trial, which caused their stock to fall. So anyone who invested in that got hit a little bit. But here's another one. There was a story not too long ago. You know, we've talked about that 23andMe and genealogy.com and a lot of the fine print that is going along with submitting your DNA and paying your $99.95 is that you lose the rights to your DNA forever and ever by feeding the cohort project. And there's a lot of ethical issues with how that data is then being used after the fact. Now, if this New York Times article from May 11th, 2018 will load, and it just sits there saying processing request, please hurry up. But they just nabbed the Golden State Killer, I believe was the name of the perpetrator, 72-year-old man, through one of his family members putting a DNA test up to one of those sites, and they were able to match the DNA sequencing through his family member and then track him back and find him after many, many years. So here's an ethical conundrum. Should law enforcement be able to use DNA from genealogy websites in criminal investigations? Well, have you or would you like to use genealogy websites like GEDmatch, Ancestry.com, or 23andMe? How concerned are you about the privacy issues and the sharing of your DNA data online? Well, in the story titled "The Golden State Killer is Tracked Through a Thicket of DNA," an expert Shudder story, Gina Colata and Heather Murphy write, quote, "Genetic testing services have become enormously popular with people looking to find their long-lost relatives or whether or not they're related to European royalty, but most never imagine that one day intimate pieces of their DNA could be mined to assist police detectives in criminal cases." Even a scientific expert applauded this week in the arrest of the Golden State Killer suspect, Joseph James D'Angelo, 72 years old, as some expressed unease on Friday at the reports that detectives in California had used a public genealogy database to identify him. Privacy and ethical issues glossed over in the public's rush to embrace DNA databases are now glaringly apparent, they said. This is really tough. He was a horrible man, and it's good that he was identified, but does the end justify the means? Well, there's a whole generation that's now saying we don't care about our privacy, and how about to add a little more insult to injury? Here's another story out of InsideEdition.com, June 1st, 2018. Astonishing new DNA technology can create pictures, human images, to help identify crime suspects. Now, you remember we read a story a long, long time ago about the artist, and she was also a biologist or something in New York City, who was collecting cigarette butts and chewing gum all around in public places, sequencing their DNA, and then doing 3D printed masks of the faces of the owners of those objects. Well, now crime fighting has taken on the same sort of capability. Identifying a perpetrator of a crime may have just become much easier thanks to a brand new DNA technology. The technology, developed by Parabon Nanolabs, has assisted police departments in 37 states and can produce an image of a person based on a simple mouth swab. Chief Scientist Ellen Gretek says that from the swab, their imagery can display information like eye color, skin color, hair color, and the shape of their face. We can predict that just from the DNA and tell investigators, here's who, and most importantly, here is who you should be and not be looking for. The technology was demonstrated by workers at the facility, and when viewing side-by-side images of actual faces and the faces based on DNA samples, they appeared to be extremely alike. So think about that one, 4 minutes. And now, to end the show in the last minute, and hopefully this story will load, because I want to give, this is the tailspin into Wednesday, the human gut virome. This is in your gut. We outline our current understanding of the human gut virome, in particular the phage component of what lives in every human's guts. Remember, we found out that phages are in every living organism, and they're also falling from the sky. The phage component of this ecosystem, we want to highlight the progress and the challenges in the viral discovery. We reveal that there are developments in high-throughput sequencing in these technologies and how the human gut virome plays a role in our health and well-being. These microbial residents have been shown to provide a wide range of essential functions, from helping us digest our food to our immune system and how they can be invading pathogens. All right, guys, we'll catch up on Wednesday. Hopefully, Clint Patrick, are you there?